In the previous section, we learned how cells divide. We learned the importance of cell division. We also learned how the cell cycle works, in that it's a complicated process that goes through, first of all, one, interphase, two, mitosis, and three, it hits the concept of cytokinesis. Today in this section, we're going to be looking at another process of cell division called meiosis. Now, before we can get an appreciation for meiosis, we need to review some key terms. First of all, sexual reproduction versus asexual reproduction. In sexual reproduction, we're going to have recombination of genetic material, whereas in asexual reproduction, this does not occur. For example, a bacteria will just divide in two. A second important term to understand are somatic cells versus germ cells. Somatic cells are basically cells of the body. The word soma means body. So all of the cells in an organism, except the cells that produce sperm and egg, are referred to as somatic cells. Now, the cells that produce sperm and eggs are referred to as germ cells. Chromosomes, we've had a little introduction to chromosomes, but chromosomes exist in pairs. One had come from the mother and one had come from the father, and paired together, we refer to these as homologous pairs. Now, all chromosomes in somatic cells exist in pairs. The concept diploid, or 2N versus haploid. A full set of chromosomes is referred to as diploid, whereas basically one of each chromosome into a pool we refer to as haploid, or basically half of the chromosomes. The term gametes refers to any cell that is involved in reproduction. Now, gametes are reproductive cells that are made in the process of meiosis, which is the subject today. Let's for a moment look at a karyotype of a human cell. This is a diploid cell. This is basically a pictorial view of all of the chromosomes that are present in all human cells. As you can see, we have 23 pair of chromosomes. Each pair is referred to as a homologous pair. As we look at this karyotype, we can see that while we have 23 pair, in reality, the pair X and Y are not a true pair. As you can see, the X is a very, very large chromosome, whereas the Y is a very small chromosome. These are the chromosomes that are responsible for sex, male or female. This shows you the disproportionate size between the X and the Y chromosome. As you can see, the X chromosome has a vastly increased amount of information compared to the Y chromosome, and this will be important to note later on. So now let's get to the main point of this lecture, meiosis. Meiosis has the total purpose of formation of gametes. The process takes place in cells that produce sperm and egg. Obviously, in humans, that would be ovaries and testes. In this process, chromosomes will go from a full set, 2N or 46, to 1N or 23. Now, the process of meiosis is very similar to mitosis, except that here we have two distinct steps, meiosis 1 and meiosis 2. Fortunately, the terminology and some of the things that take place are very similar. For example, in meiosis 1, we refer to prophase 1, metaphase 1, anaphase 1, and telophase 1. In meiosis 2, we refer to the steps involved here as prophase 2, metaphase 2, anaphase 2, and telophase 2. So now let's look and review this overall process. Remember, meiosis' total purpose is to make sperm and egg. As we look at this cell to your left, we see that this cell is in the process of finishing off interphase. The DNA has already duplicated, and this cell is destined to form either a sperm or an egg. As you can see, the first thing that happens is you go through the process of prophase. In this case, we are looking at two pairs, homologous pairs of chromosomes. As you can see, in this phase, the pairs get together, basically hug each other, and go through a process called crossover or synapsis. In this process, genetic material is exchanged between homologous pairs. Now we go to metaphase 1, and here we see the chromosomes line up. But unlike in mitosis, you can see the chromosomes line up as homologous pairs rather than in a single line across. The homologous pairs in anaphase, as you can see, are now moving apart to their respective poles. And finally, in telophase, we have two new daughter cells produced. But this is just the end of meiosis 1. Now, what has happened here is a reduction division, in the sense that we started out with 4 chromosomes, and now we end up with 2 chromosomes per cell. We have gone from 2N to 1N. Now, as you can see, now you can see that in meiosis 2 is essentially identical to mitosis. The 2 chromosomes will now go through a process of prophase 2. In metaphase 2, the single chromosomes line up, and the chromatids now get pulled apart, and you end up in anaphase 2 with all of the chromatids moving towards their respective poles, and the end result from meiosis 1 and meiosis 2 are 1, 2, 3, 4 gametes. And notice that we started with 4 chromosomes, and we end up with cells that have 2 chromosomes. So therefore, we have gone from 2N to 1N in the process of gamete formation. Also notice that every one of these gametes are unique, and that's an important point to note. In the process of meiosis, an important concept evolves. And this concept is that you get genetic variation. So what is the source of genetic variation in gamete formation? The first source takes place in prophase 1 when synapsis or crossover takes place. The second source of genetic variation takes place in metaphase 1, where you have independent assortment. And we'll talk to each of these in a moment. And finally, mutations and a variety of chromosomal abnormalities may take place during the process of meiosis and, of course, gamete formation. All right, let's look at the first source of genetic variation. This took place in meiosis 1 when crossover took place. As you can see, we started off with 2 chromosomes. These are homologous pairs you see to the left, and each pair has 2 sister chromatids. During this process, the homologous pairs, as you can see, come close together, and they basically go through this hugging, they go through this dance so close to each other that allows material from one homologous pair to jump over to another homologous pair, and look what you end up with. You end up with two new chromosomes that are somewhat different from the original two that you started with. And this is, again, the first source of genetic variation. The second source of genetic variation takes place during metaphase 1 of meiosis. In metaphase 1 of meiosis, we can see that the homologous pairs line up, one on the opposite of the other. If we can picture, now here we're looking at just 4 chromosomes and 2 pairs. However, in the human, we have 23 pairs. So what is to determine which pair goes where? This is an independent assortment process, so that every time in metaphase 1 these pairs line up, an entire new set of gametes will result. As you can see on the first example of metaphase 1, this is the way these chromosomes line up. However, on the second example of metaphase 1, we can see that the shuffling of chromosomes are a little bit different. Because they are different, we see that the 4 gametes that will be produced will be quite different from the 4 gametes that were produced in the previous shuffling that took place in metaphase 1. So now that we understand the process of meiosis, let's just kind of review and look at what happens during spermatogenesis, which is the formation of sperm, and oogenesis, which is the formation of eggs. In spermatogenesis, we can see that we're starting off with 4 chromosomes. One, remember, has come from each parent. Okay? The 4 chromosomes now, as you can see, during the primary spermatocyte have become, each has formed 2 new chromatids, and they've now lined up during meiosis 1, and reduction division has taken place, so we've gone from 4 chromosomes to 2 chromosomes in the secondary spermatocytes. This process now goes through meiosis 2, and 4 new little tadpoles, sperms, have been formed. As you can see, we've gone from 2N to 1N, from 4 chromosomes per cell to 2 chromosomes present in each sperm. In oogenesis, the same process occurs, but notice that we end up with basically 1 egg and 4 polar bodies. That's just the way nature works. So that the efficiency of spermatogenesis obviously is much more than oogenesis. So now that we understand how sperm and eggs are formed and how gametes are formed, basically, remember the sperm is 1N, the egg is 1N, and during fertilization, sperm and egg combine, and the first thing that is formed is a zygote. And we're going back to 2N again. The zygote, of course, is the first little structure that eventually leads to the development of a full organism. Just to give us a little appreciation of how males and females are formed, it's quite simply. The father determines the sex of the offspring. If you look at the mother and the father, and you look at the mother has 2 X's, which of course indicate female. The father can only have an X and a Y. During the process of meiosis, the only gametes that can be formed by the mother are going to be X's, X's, and X's. However, the father produces 50% X's and 50% Y's. So during the process of fertilization, there's a 50% chance that's going to be a son or daughter. But who contributes the Y chromosome? It has to be the father. Therefore, the father determines the sex of the offspring. Now, this slide reviews mitosis versus meiosis. I won't spend a lot of time on it, and you can look at it carefully yourself. But basically, mitosis is a process for cell division, for cell growth, cell development, repair, and so on. Look carefully at mitosis. We started off with 4 chromosomes, which is 2N, a full complement. And what do we end up with? 2N, identical new cells with the same number of chromosomes. Whereas in meiosis, we start off with 2N. In meiosis 1, we go through a process of reduction division. We go from 2N to 1N, so that all of our gametes end up with half of the genetic components. So we start with 2N, we end up with 1N. So all gametes have half of the genetic components. Now, there are some other processes that do not involve recombination of genetic material. We refer to this as asexual reproduction. Parthenogenesis takes place in some animals. In parthenogenesis, basically, an egg will develop into a full organism without any fertilization whatsoever. In binary fission, which takes place in bacteria, an organism simply divides itself in two. And in regeneration, an organism is able to basically reform a given appendage or even a completely new organism. Now, let's look at reproduction by way of parthenogenesis. This example shows you a lizard that lays eggs, and the eggs can spontaneously develop into a complete new organism. Therefore, the entire population of this species, yes, are going to be all females. So therefore, males can be irrelevant in a given species development. Now here we have a starfish regenerating. If you cut an appendage off of a starfish, the organism can regenerate into a completely new species. Now this should not surprise us. Remember, every cell in every organism has all of the genetic material to form a complete new organism. Reality is, in cloning, we can take a cell from an organism and make it into a completely copy of the organism it came from. The only reason this doesn't happen in all of our cells is that the genetic material for development is basically shut off, and a cell will only use the information necessary to carry out its particular task.